Hi there, all you biology fans. Mr. Mechnick here with another podcast on cellular respiration. Yes, I know this is the one you've been waiting a long time for, so here it is. So today's purpose is to explain how your cells can harvest energy from the food that you eat. And I would like all of you to be able to describe what happens during each one of these chemical pathways: glycolysis, the Krebs cycle, and the electron transport chain. So how do we get energy from the food that we eat? A good way to help you organize this podcast is probably to make a chart similar to this one. And the chart will do a comparison of glycolysis, the Krebs cycle, and the electron transport chain in aerobic cellular respiration. So we will look at these processes, and you should be able to recognize if oxygen is present. We should be able to look at each one of these pathways and identify what is the starting product, what goes into the reaction, and then what are the end products, what comes out of the reaction. And then ultimately, why is this process important? How do we use this in the body? How many ATP are produced during each one of these steps? And then where in the cell does it happen? So this chart would be a nice way to organize the information presented in this podcast. So take a minute and copy a chart like this into your notes, and then strap it on and let's get into it. So as we know, glucose is this very important biomolecule that our cells require for energy. So just to give you a little idea, a single glucose molecule provides about 686 kilocalories worth of energy. And it sounds like a lot of energy, but our cells really can't utilize that at all. So we have to convert it into something more useful, like adenosine triphosphate or ATP. So we have to use many different chemical steps in order to make ATP. And for every ATP molecule, we only get 7.3 kilocalories. So it seems like, why couldn't we just use this to get our energy instead of ATP? Well, over time, cells aren't able to process this large complex molecule, where ATP becomes a good donor molecule of phosphates to drop off energy to our cells so our cells can provide their work. So in reality, our cells really aren't the most efficient at converting glucose into ATP. 38% of the energy from a glucose molecule is utilized in the form of ATP. That means 62% is a byproduct or is a waste product in the form of heat. So here's a general overview of the process that I'm going to discuss today. Glucose will enter our cells and go through a process known as glycolysis. This is the first step. It happens outside of the mitochondria in the cytoplasm of a cell. The products of glycolysis are going to be transported across the membranes of the mitochondria, and within the mitochondria we're going to see the Krebs cycle occurring. And the Krebs cycle is a process that uses a pyruvic acid, and it makes some ATP and will send a bunch of electrons to the electron transport chain, along with some hydrogen ions that are carried by these NADH and FADH2 molecules. So this right here is what we're going to focus on today. So glycolysis simply means splitting sugar. So we all know that glucose has 6 carbon atoms, and when we split a 6-carbon glucose molecule, we end up with 2 pyruvic acid molecules, each one containing 3 carbons, and it also generates some ATP as a result. We have to invest a little energy to get this glycolysis process to work, but lo and behold, we end up with 2 ATP that our cells can utilize to do work. Oxygen is not required during glycolysis, so this is actually an anaerobic process because we don't need to invest oxygen to split glucose. And like I said earlier, it occurs in the cytoplasm of our cell. So when we have plenty of oxygen available, cells will go through aerobic respiration. So that's the purpose of this presentation today, is to go through aerobic processes. So aerobic requires oxygen. I just said glycolysis does not. So this is an anaerobic pathway, but we need to split apart glucose into these 2 3-carbon chains, which are called pyruvic acid. You got it. So when we produce that ATP as a result of converting glucose into pyruvic acid, we also have to invest some ATP. So think of ATP like a charged battery, has lots of potential to do work. And when we use a battery up, it loses its charge, and the uncharged ATP molecule is not called adenosine triphosphate. It's now called adenosine diphosphate. But we know that the ADP has the ability to recharge itself simply by grabbing more phosphates. And where do we get those phosphates from? Exactly, from the foods that you eat. So we can refer to ATP as this energy currency. It's 100% renewable. When we drop off a phosphate and our cells utilize the energy stored in that chemical bond, we're going to be able to do lots and lots of work with ATP. All the extra adenosine diphosphate molecules then become really good acceptors of phosphates, and we can pick up those extra phosphates from the food that we eat to build more ATP. So there's a constant cycle of ATP to ADP. As our cells require it, we break apart this bond and make more ADP. And so it's a constant cycle. We consider ATP as currency because we can spend it, we can store it, or we can build more of it. So how is ATP utilized in our cells? So here's a couple general things that cells use ATP for. We can use it for growing. We can use it for making new cells and reproduction. We can use it to transport materials in and out of cells. So materials can move across cells, and sometimes we have to expend energy to do that. In order for your neurons to fire and to transport sodium and potassium ions across their membranes, we have to expend a lot of ATP to do that. To make muscles contract, to make cilia move, to make a flagella on a cell move, you have to expend energy to do that. So for mechanical work to happen, we have to invest ATP. But the reward is great because it gives us opportunities to do lots and lots of activities. So once glucose is broken down into pyruvic acid, that pyruvic acid can diffuse across the membrane, the outer membrane of the mitochondria, go through the inner membrane of the mitochondria into what's called the matrix. So the matrix is the area in between all the folded membranes called the cristae, and within the matrix, this is where pyruvic acid is going to be converted and torn apart to produce carbon dioxide. So this is the Krebs cycle. The Krebs cycle makes carbon dioxide as a byproduct, and it also generates some high energy electrons that can be transported with the help of NAD plus and FAD plus. Basically, they're going to pick up hydrogen ions and they're going to help transport these high energy electrons to the electron transport chain. The Krebs cycle is a very important step in the process. It does yield 2 ATP as a result. And this again provides some energy for our cells to do work and happens in the mitochondria. We are not going to get into the details of the process of the Krebs cycle. You just need to know the Krebs cycle takes in pyruvic acid, it produces carbon dioxide and some carrier molecules, and that's represented by all of these yellow boxes around those high energy molecules that are going to be transferred to, you got it, the electron transport chain. So when we get all of this information carried by the NADH and the FADH2, it's going to arrive at the inner membrane of the mitochondria. So we can see the inner membranes right here, and the inner membrane is going to process and utilize the NADH and the FADH2. So it's really using the energy from these transport molecules to start creating concentration gradients of hydrogen ions. And as a result of this, it allows for these electrons to be pumped along a transport chain, and ultimately, by building up a bunch of hydrogen ions or protons on the inner membrane space of the mitochondria, it's going to create a gradient, and a gradient means that there's going to be more hydrogen on one side of the membrane compared to the other side, and eventually these hydrogen ions are going to want to travel from a higher concentrated area to a lower concentrated area, and they're going to travel through this very, very cool enzyme called ATP synthase. So there's a ion channel that will allow for hydrogen ions to pass through. It will activate this enzyme, and as a result of this enzyme activation, it will allow ADP to pick up a bunch of phosphates and build ATP like no other. It is so important for your cells to do this because as a result, you're going to make lots and lots and lots of ATP for every single glucose that enters into this process. So you start with these high energy electrons in the form of NADH and FADH2. They're going to flow down that electron transport chain, and ADP is going to be converted into ATP, and we're going to make lots and lots of ATP during this process. And it all happens because there is going to be a transportation of hydrogen ions across a gradient. And when it's all said and done, with the electron transport chain, your cells will be able to produce 36 adenosine triphosphate molecules. Yes, that is correct, kids. 36 adenosine triphosphates for every single glucose molecules that enters into a cell. Wow. That is amazing. 36 ATPs. So when it's all said and done, here's what you have. You have glucose that enters into the cytoplasm of a cell, gets split into pyruvic acid. That pyruvic acid is going to enter the Krebs cycle. The Krebs cycle is going to generate lots of carbon dioxide, and it's also going to transport all of these high energy electrons with these useful carrier molecules, NADH and FADH2. These electrons in the hydrogen ions get transported to the electron transport chain. That happens in the intermembrane space of the mitochondria, and as they get transported along, they will create a gradient, creating lots and lots of ATP. So when you combine the two ATP made by the Krebs cycle and the 34 produced by the electron transport chain, you're getting 36 ATP going through the aerobic process. And I forgot to mention, during the electron transport chain, this is where oxygen is invested. So O2 plays an important role in aerobic respiration, and this is where we have the investment of oxygen. So this chart is a nice chart that might be useful to help you fill in that initial chart to compare the three different processes of aerobic cellular respiration: glycolysis, Krebs, and the electron transport chain. Stay tuned because sometimes glycolysis will occur and split into 2-pyruvic acid, but if there's no oxygen present, then your cells must go through a different process. Yes, there's more to know. And stay tuned for another podcast on the anaerobic pathways that happen in living cells. Anaerobic means that there's no oxygen. But again, this is a simple diagram. Maybe it's not so simple, but it's a nice overview of the process of how your cells make ATP so you can do all of the work that you do every single day. Thank you, and if you have not subscribed to this channel, please do so. What are you waiting for? Subscribe now. See you in class tomorrow.